This is the video for Lab 9, Disk Diffusion. The purpose of this lab is to understand the process of a disk diffusion assay, be able to interpret disk diffusion results, and to be able to interpret Kirby Bower results. There are many different types of antimicrobial substances, and some examples are iodine, hydrogen peroxide, phenols, like you find in Lysol, and hand sanitizer. And these different substances can be used on different surfaces, such as iodine, hydrogen peroxide, and hand sanitizer can be used on skin, while Lysol is used on surfaces. And these different substances can have different effect on the bacteria. One way to test the effectiveness of different antimicrobial substances is to perform a disk diffusion assay. In lab, you will be performing a disk diffusion assay to test the effectiveness of 4 different antimicrobial substances. So to perform this experiment, the first thing you will need is a plate, a TSA plate, and you will inoculate that plate with a particular type of bacteria. To inoculate the plate, you will use a loop, and using the loop, you will streak all over the surface of the plate. The purpose of this type of inoculation is to spread bacteria all over the surface, and you can even go perpendicular, aseptically get more bacteria and go perpendicular, but you want to create a lawn of growth. And a lawn of growth means that the bacteria is grown in a nice even layer over the entire surface of the plate. Once you have inoculated the plate, then it's time to add the discs. And each disc has been soaked in one of the 4 different types of substances you are going to test. So you will use forceps, tweezers to place each disc on the plate over the lawn of bacteria. And the idea behind a disk diffusion assay is that during incubation, the chemical that has soaked into the disc will diffuse through the agar, and if that chemical is able to inhibit the growth of the bacteria, then there will be a zone of inhibition around the disc. So when you look at the plates after 48 hours of incubation, you should observe nice even growth. So the yellow is the even growth all over the plate. And then you need to look for zones of inhibition. A zone of inhibition, again, is a clear area where the bacteria were unable to grow. And some zones of inhibition may be very large, like this one, and some may be very small. And you may have absolutely no zone of inhibition where the bacteria were not inhibited at all, and they grew all the way up to the disc. To analyze the zones of inhibition, you are going to measure them in millimeters. So the ruler is numbered in centimeters, but you should know that there are 10 millimeters in every centimeter. So if it measures half a centimeter, that would be 5 millimeters. And you will be measuring the diameter of the zone of inhibition. The diameter goes all the way across from one edge of the circle to the other, crossing through a center point. These are some potential results you could see from a disk diffusion assay. And in this example, a student tested 4 different types of bacteria, so bacteria type A, type B, type C, and type D, and tested 4 different antimicrobial substances. So substance 1 was tested on all the types of bacteria. And what you should notice is that different antimicrobial substances can have different effects on different types of bacteria. So for example, substance 3 with organism A had a fairly large size zone of inhibition, but when substance 3 was tested on bacteria C, it had a very small zone of inhibition. So the different substances can affect different species of bacteria differently. Antibiotics are an unusual type of antimicrobial substance because they are derived from different types of fungi and bacteria, and their purpose is to target bacteria and inhibit the growth of bacteria. And this is because the microbial world is a very competitive environment, and if a fungus or a bacterium can secrete a substance to kill or inhibit competitors, that will give the original fungus or bacteria a selective advantage. So there are hundreds of different types of antibiotics, and they can target different parts of the bacterial cell. Some can target the cell wall. Others can target metabolic processes. There's some that can target the cytoplasmic membrane, others DNA replication, and there are others that target transcription, and still others that can target different aspects of translation. So there are many different types of antibiotics, and they can have varied effects on different types of bacteria. For example, these antibiotics that have an effect on cell wall synthesis are usually much more effective on gram-positive bacteria compared to gram-negative bacteria. When performing a disk diffusion assay on antibiotics, it is called a Kirby Bower. And the Kirby Bower assay is performed almost exactly like a regular disk diffusion assay. The first thing is to create a lawn of bacteria. So creating the lawn of bacteria is the same, but the media will be slightly different. So instead of using TSA when performing a Kirby Bower, use Mueller-Hinton agar. But creating the lawn is the same. Using a loop, you will streak all over the surface of the plate, again trying to spread bacteria everywhere so that you will get even growth. And again, aseptically, you can streak the other direction. Once you have streaked all over the surface of the Mueller-Hinton agar, then it is time to add the discs. And for a Kirby Bower, there is a specially made dispenser that will dispense 8 discs evenly right on the plate. And all of these discs are labeled so that you know which antibiotic is in which disc. This is an example of a Kirby Bower plate after 48 hours of incubation. And as you can see, many of the discs have a zone of inhibition. So here's a zone of inhibition. And again, you will measure the diameter of the zone of inhibition. So the diameter goes straight across the zone, crossing through a center point. Sometimes the zones of inhibition will be so large on the Kirby Bower that you won't be able to see a complete diameter. And if that is the case, you need to take a center point from the disc and measure 1 radius. And if you multiply that by 2, that will give you the measurement for the diameter. And again, the measurements need to be in millimeters. So 1 centimeter equals 10 millimeters. Now, another thing that is different about the Kirby Bower is that the relative size of the zone of inhibition does not necessarily tell you how effective the antibiotic is. In order to determine if the antibiotic is effective on a particular strain of bacteria, you need to use this chart. So after you have measured the zones of inhibition, you need to look up the size on the chart to determine if the bacteria is resistant, intermediate, or susceptible to the antibiotic. And with some of the antibiotics, like ampicillin, it depends on the type of bacteria. So for instance, Enterobacteriaceae, that would be like an E. coli. If the zone is smaller than 13 millimeters, it is resistant. If it is larger than 17 millimeters, it is susceptible. Whereas for Staphylococci, if it's smaller than 28 millimeters, it is resistant. So in order to accurately interpret the results of a Kirby Bower, you must look up the results on this table to determine how the bacteria have responded to the antibiotic, if they're resistant, intermediate, or susceptible.